First, I would like to thank the chair of the BSN for giving me this opportunity to talk about my research with you all. And today I will talk about an in vitro model that we have optimized to study synaptic connectivity and how it changes under pathological conditions. Now, as you all know, information processing in the brain relies on the constant changing of neuronal connectivity within and between these very complex neuronal networks. And this information processing, this information processing is affected in disorders like schizophrenia and Alzheimer's disease. Now, the main focus of my research has been to see if we can model the synaptogenesis and synaptic connectivity in a fairly simple in vitro model, because such model would be very interesting to study the mechanisms behind neurodevelopmental and neurodegenerative disorders. To this end, we're using primary hippocampal cultures which we make from mouse embryos. We're using these cultures because they're known to recapitulate neuronal network formation in vitro. As you can see here, between 0 and 7 days they form this interconnected neuronal network. I plate these cells in 96 well plates because I want to extract a maximum amount of data with a fairly small amount of biological material. And another advantage of this approach is that these cultivation conditions are very tightly controllable, and that these neurons are easily accessible for pharmacological and genetic interventions. Now, I've been optimizing three main readouts for neuronal connectivity, being neurite outgrowth and synapse density, dendritic spines, and a functional parameter, electrical activity via calcium imaging. Now, neurite outgrowth is quantified by measuring the surface area of a beta-3 tubulin staining, while synapse density is expressed as the number of these synaptophysin positive puncta per square micrometer of tubulin. And when we do this on consecutive days, we see that neurite outgrowth increases until 11 days in vitro, while synapse density increases up to 14 days in vitro. Now, a second morphological parameter are the dendritic spines, which are these tiny protrusions that emerge from the dendritic shaft. And each of these spines contains an excitatory synapse, and the morphology as well as the density are correlated with the synaptic strength, and they're also quite motile, as you just saw in this movie. So manual quantification of these kind of images learned that the synapse density is mainly increasing in later stages, so this corresponds to the fine-tuning of the neuronal network. Now, we're also aiming to quantify these spines in a more automated fashion, but an often recurring problem in automated spine quantification is that these images are very full of neurites, which makes it quite hard. And one strategy to overcome this problem would be to transfect all of the neurons with a photoconvertible protein and then do the conversion just in one neuron, so that you end up with one neuron in your field of view. A third parameter, and this is the functional parameter, is electrical activity. And here we see that between 5 and 7 days there's a clear maturation, because at 5 days in vitro we have this non-correlated activity, while at 7 days in vitro you see these nicely correlated network bursts. And these kind of calcium recordings are quantified by a MATLAB image analysis that we have developed that recognizes neurons based on a nuclear image and the response to a high concentration of glutamate. And then it can extract different parameters both on the single neuron level and on the neuronal network level. Examples of such parameters are the bursting frequency, the bursting amplitude, and the decay time at the level of the single neurons. And on the neuronal network level, we see a marked increase in the percentage of active neurons, the frequency of synchronized bursts, and the correlation score, which tells something about the synchronicity of the signal between individual neurons. And we see this increase between 5 and 7 days in vitro also. Acute pharmacological perturbation showed us that these calcium oscillations are directly related to electrical activity because they can be stopped by TTX. And this synchronized network activity is mediated by the NMDA receptor because an NMDA receptor antagonist can decrease the correlation score. Next to fluorophore as a calcium indicator, we're also looking now at a genetically encoded calcium indicator, which has several advantages over fluorophore. For example, its kinetics are faster, like you can see here. It allows for long-term recordings, and from an image analysis perspective, it's easier to use because the neuronal the expression is neuronal specific, and the nuclei of the neurons only are also visible in the red channel. So now that I've showed you the three parameters that I use to quantitatively describe neuronal network connectivity, I will show you how we use them to study the effect of microtubule stability on neuronal connectivity. So neuronal microtubules are very important for neurite outgrowth and also for the transportation of cargo from the cell body to the most distant synapses. And recently it has also been shown that these microtubules, that there is a direct crosstalk between them and the synapses. Now, we saw that when we administer a single dose of Taxol at 4 days in vitro, that you have a negative impact on both neurite outgrowth and on synapse density. And also the amount of dendritic spines is reduced, and there are some defects visible in the electrical bursting pattern of these Taxol-treated neurons. On the other hand, nocodazole is a compound that does quite the opposite of Taxol, so it impedes free tubulin to assemble into microtubules. And when you treat the cultures for 4 hours, you see that this microtubule network is partly degraded after 4 hours. And this has an impact on the electrical activity, as you can see here. And the decreased synchronous bursting frequency can be rescued by adding stabilizers after 2 hours. So to conclude, I can say that we have validated an in vitro platform for pharmacological and genetic interrogation of synaptic connectivity, which is accessible, amenable to upscaling, so it could be used, for example, for neurotoxicity screening or target identification of phenotypic drug screening. And now for the future, we're interested in human iPSC-derived neuronal cultures to see if we can use the same readouts for connectivity in those kind of cells. And I'm currently also looking at the effect of tau aggregation and spreading on neuronal network connectivity. So I would like to thank my colleagues of the Lab of Cell Biology and Histology and the colleagues of Janssen Pharmaceutica. And of course, I thank you all for your attentions. Thank you. Calcium imaging gives some information about the connectivity of the network, but to correlate this to the human situation, it's a bit difficult.
No, 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 I'm not asking about the human. I'm asking about the, because in vivo, because quite a lot is known about the hippocampal development in the mouse, just from the acute slices, for example, or in vivo recordings. Can you correlate the timing, what you see in the culture, to what is shown in vivo or in vitro using the slices?
Well, I'm not sure when these giant, the time point when these giant depolarizations are starting to happen in slices, but yeah, I can say that between until like 5 days up to 7 days in vitro, you see the correlation of the network activity, and later on you see more the fine-tuning of the synapses, so spines emerge like you would see in the hippocampus, and also the bursting pattern changes a bit. There is more, it's less yeah, they fire on a less constant rhythm. So.
Questions? I have one question and one comment. You make me feel a little bit younger because we used to work with neuronal cultures, I would say 20, 25 years ago. And I was wondering, what do you think about the neurons that are present in the culture? Because we know that at least in the hippocampal network, in vivo or in slices, you get different cell types. What are the neuronal cell types that you are studying here?
Yeah, I think they're mainly pyramidal neurons. And of course you have some glutamatergic and GABAergic neurons, because it's very important that the mix is right, otherwise you would not see this synchronization of the activity, because also GABA is at first excitatory, and then until like between 5 and 8 days in vitro it will switch to an inhibitory phenotype. So yeah.
Did you look for inhibitory activity as well, or?
Yeah, so
GABA?
when you add GABA inhibitors, then you start to see the normal reaction, so increase of the spontaneous bursting activity only in cultures which are older than 8 days in vitro.